Alright, essay questions for Unit 2 exam. First one. Describe the molecular characteristics of the contractile filaments found within the functional unit of the skeletal muscle cell, the sarcomere. Describe the mechanism by which an action potential in the sarcolemma results in release of calcium from the sarcoplasmic reticulum, followed by a description of the molecular mechanisms of muscle contraction. Discuss the various sources of energy utilized for muscle contraction. Include the relative amount of each source available to the cell, the advantages and disadvantages of each type, and under what conditions each might be used. Alright, the contractile filaments of the sarcomere are the myosin fibers, and the general sarcomere is repeating units like this, and myosin is this central band. And then myosin fibers are shaped similar to golf clubs, long shafts that curl together to make up the A band here in the center. And the head of the myosin is hinged at its base. This motion is what creates the power stroke of the muscle contraction. And at the head of the myosin is where it attaches to actin to move it. So once an action potential reaches the end of a neuron, once it reaches the end of a neuron that connects to a muscle fiber, acetylcholine is the neurotransmitter that is used to communicate with skeletal muscle. And the acetylcholine receptors of the muscle cell are in the region known as the sarcolemma, which is also known as the motor end plate. So acetylcholine binds to ligand-gated sodium and potassium channels, which depolarize sarcolemma. And their depolarization also activates voltage-gated sodium channels. And these channels propagate the action potential throughout the neuromuscular junction. And as the action potential moves around the cell, encounters and activates dihydropyridine channels, which are voltage-gated calcium channels. But these channels are not used to bring in extracellular calcium. All calcium used by muscle cells. All calcium used by muscle cells comes from within the cell. Instead, this channel is linked to a ryanodine receptor, and these open to allow calcium to exit the sarcoplasmic reticulum here. This holds all of the calcium necessary for the muscle cell. So dihydropyridine channels only act as a voltage sensor to activate the ryanodine receptor channels. And so once this is activated by the action potential, opens the ryanodine receptors, and the calcium in the sarcoplasmic reticulum can exit, cause muscle contraction. So once calcium is released, it is able to bind onto the sites on troponin C to remove tropomyosin from actin. So back here, actin, out here. Its active sites are covered by tropomyosin. Calcium attaches and removes tropomyosin so that myosin can attach. And at this point, myosin heads are in the cocked position, like that, and have an ADP and phosphate on them. And as myosin and actin form a bond, energy is released, and some of that energy is recaptured into myosin's phosphate here, which breaks it off, and the rest of the energy is lost to heat. And the loss of that phosphate group causes the myosin to have a conformational change and move to its uncocked position. And so uncocking pulls actin with the myosin head, and multiplied by a few million of these myosin heads causes the total length of the muscle to shorten, causing a contraction. This is known as the power stroke, and the ADP is also broken off during this power stroke. And in order for the muscle to shorten a significant amount, the myosin head must detach and reattach further down along the actin filament, like climbing a ladder. And in order to detach myosin from actin, a new ATP molecule must be brought in. And the formation of the bond between ATP and the myosin head releases energy, which will be used to break the bond between actin and myosin. And once separated, the myosin head will hydrolyze the ATP back into ADP and another phosphate group, which causes the head to become cocked again. So ATP is attached to break myosin and actin apart, and then myosin hydrolyzes that ATP into another set of ADP and phosphate in order to move into its cocked position again. And so for sources of this ATP, the first source is simply free ATP that floats around in the cytoplasm. This is a very small supply, and this will be used up in 1 to 5 seconds. And then creatine phosphate is the second source. Creatine phosphate readily gives its phosphate group to ADP and recycles it back into ATP without the use of oxygen. The supply of creatine phosphate lasts for around 10 seconds. Next is glycolysis. The glucose used for this comes from stores kept within the muscle and can fuel the muscle for about a minute. And the last energy source for muscle is oxidative respiration, which makes use of the mitochondria within the muscle cell. This process is much slower at producing ATP than the previous methods, since it's aerobic and the others are anaerobic. But this method will fuel the muscle for hours on end. But being a slower process, it can't supply the muscle under intense activity. But for things like jogging, it can readily supply the muscle for hours on end. Alright, second question, describe the two main types of smooth muscle. Describe any molecular mechanisms of muscle contraction in smooth muscle. Begin at the point of calcium entry and end at the point of contraction. Compare and contrast smooth and skeletal muscles structurally, functionally, and regulation-wise. So the two main types of smooth muscle are single unit and multi-unit. Single unit smooth muscles are connected by gap junctions, which allow the muscles to contract as a single unit. So lined up side by side, these single unit muscle cells have channels linking each to each other. And so if a depolarization is caused here, that flood of this change in depolarization can travel through here, activate it here, travel through here, activate it here. And the contraction moves through the muscles as a wave. This contraction within the digestive system is called peristalsis. These types of cells depend on certain cells to automatically depolarize to start the contraction. So these muscles can contract independent of the nervous system, but can be modified by it. The other type of smooth muscles are multi-unit muscles. These muscles are not electrically coupled, so contractions do not spread on their own. They rely on input from the nervous system or the endocrine system. Examples would be the iris and the erector pili muscles in the skin that control hair. So for hair to stand on end, it's caused by these erector pili muscles, and they're individual for hair, so being linked wouldn't make any sense. So they're part of the multi-unit smooth muscles because they're not consciously controlled. So calcium used in smooth muscle can come from intracellular sources or extracellular sources. It's one of the big differences between smooth and skeletal. Skeletal stores its calcium within the sarcoplasmic reticulum. This is the only calcium it uses. But there is still a massive amount of calcium outside of the cells and the extracellular fluid. And so smooth muscles can make use of this, though they still have their own stores. Some use either or both. So once calcium has entered the cell of a smooth muscle due to an action potential or ligand, it binds to a protein unique to smooth muscle called calmodulin. And since smooth muscle does not utilize tropomyosin to cover active sites on actin, calmodulin is activated when calcium is bound to it, and once activated, it will activate another enzyme called myosin light chain kinase by exposing a previously covered active site. So that myosin light chain kinase sort of wraps around itself to cover that active site. But once calcium comes in and activates calmodulin, activates myosin light chain kinase, and exposes its active site, activating it. Then myosin light chain kinase will phosphorylate the regulatory light chain of the myosin molecule. So starting out the same with ADP and phosphate, but this myosin light chain kinase will add another phosphate down here in order to activate the myosin. And this phosphorylation will increase the affinity that myosin has for actin. And this step requires its own ATP, separate from the one used for caulking the myosin head. And once phosphorylated, the myosin will be able to attach to actin once this phosphate is added. So ADP and its phosphate group will then dissociate to allow the myosin head to do its power stroke. So this phosphate stays behind. First, ADP and phosphate are removed, and myosin goes through its power stroke. And ADP then attaches to the myosin head to disconnect it from actin. So ATP comes back in order to break the two up. And is still in its decocked position with just the ATP, but it is hydrolyzed back into ADP and phosphate in order to recock. But as long as the regulatory light chain stays phosphorylated, the myosin head is ready to attach to a new active site. Phosphate. So as long as this remains here, it's ready to begin the cycle again without having to get more calmodulin, more myosin light chain kinase. As long as that stays, it jumps right back here. But then if myosin light chain phosphatase is available, then it will remove this phosphate on the light chain and lower myosin's affinity for actin, preventing it from attaching to a new active site and causing the muscle to relax. So smooth and skeletal muscles are different in their methods of regulating the attachment of myosin to actin, as well as their sources for calcium. On to question 3, first describe and explain, and then compare and contrast the muscle sensory receptors, muscle spindles, and Golgi tendon organs. Describe the stretch reflex and the withdrawal reflex. And part 2 of this essay described the chemoreception of smell and taste. Finally, describe how energy carried by sound waves turns into mechanical vibrations, fluid waves, chemical signals, and finally action potentials for the things we hear. So muscle spindles are meant to respond to changes in muscle length. So muscle spindles are embedded directly in the muscle fibers as sensory neurons. And there are 2 types of spindle fibers, and they respond to stretch differently. Bag fibers with type 1A sensory innervation are most sensitive to the rate of stretch, and chain fibers with type 2 sensory innervation are slow to respond and are sensitive to the amount of stretch or the length of time the stretch is occurring. And these spindles cause the stretched muscle to contract in order to prevent injury from overstretching. So separates muscle from tendon, and then the Golgi tendon organs are sensitive to the tension within a muscle. So their neurons reside directly in the tendons of muscles. So muscles have the strength and power to rip their tendons or tear off pieces of bone at their insertion points. So the Golgi tendon organ's job is to detect when tension from a muscle contraction is becoming too strong for the tendon or bone to hold together. And in response to a Golgi tendon's activation, the muscle is deactivated and forced to relax. So the stretch reflex involves the spindles detecting an excessive stretch and stimulating its muscle to contract, and its opposite to relax. And the withdrawal reflex involves a Golgi tendon detecting excessive tension within a muscle and its response of relaxing the tense muscle and activating its opposite to relieve tension. So part 2, chemoreception of smell and taste. So for taste, an action potential is created by a taste bud when certain tastants enter the cell and cause a depolarization of the membrane, allowing voltage-gated calcium channels to open, activating vesicles to release neurotransmitters into a synaptic cleft for a neuron. And different taste buds allow different ions to enter depending on what type of flavor they detect. So taste bud here, and so there's salty, sour, sweet, bitter, and umami. So salt, the perception of salt depends on sodium. And sour relies on hydrogen ions. And the sweet, umami, and bitter receptors detect a handful of different molecules, so they make use of G proteins to detect the different molecules dissolved within saliva. And so these being positive ions already, when a food with these ions dissolves in the saliva and those ions are free to move in. They cause a depolarization of the membrane, activating voltage-gated calcium channels, and the calcium that enters activates snare proteins that release vesicles with neurotransmitters that activate an action potential in a neuron. Then for the G proteins, for sweet, bitter, and umami, once it's activated by its certain solvent, it releases a secondary messenger that opens calcium channels on the cell surface or on the smooth endoplasmic reticulum to increase calcium levels. So for smell, odors must dissolve into the water-based mucus to reach the membranes of receptor cells, where they will bind to the cilia. Olfactory receptors use G proteins, which activates the enzyme adenylyl cyclase when stimulated. And the activated adenylyl cyclase will then produce cyclic AMP, which opens sodium and calcium channels to depolarize the cell. So, up in the nose, so odors need to dissolve through the mucus, where if a certain odor binds to a G protein, it activates, and when activated, they upregulate adenylyl cyclase, which produces cyclic AMP, which comes over to channels to bring in sodium and calcium, cause depolarization, and this itself is a neuron, and so the depolarization caused by the entrance of these ions will propagate. That's how smell works. So for the third part, how energy is carried by sound waves and turned into action potential. So sound first enters the ear, and so in the outer ear, travels down the canal and strikes the tympanic membrane. And from there it enters the middle ear. And in the middle ear there are three bones, malleus, incus, and stapes. So malleus, incus, stapes, and these three all amplify the sound and vibrate against the oval window, which creates waves in the fluid contained within the cochlea, and this oval window represents the transfer from middle ear to inner ear. And the cochlea is divided into these three parallel chambers. The scala vestibuli, which which is connected to the oval window, the scala tympani, and the cochlear duct. And the cochlear duct contains endolymph, which resembles intracellular fluid. So it has a very high potassium concentration, and potassium, as opposed to calcium, is the main ion that causes action potential depolarization for this sense. And the spiral organ that converts mechanical vibrations into electric potentials is on the membrane that separates the cochlear duct from the scala tympani. So we can blow this up a little bit more. And so this membrane can detect different frequencies by being short and stiff near the oval window and becoming longer and looser as it goes further in. So different areas resonate with different frequencies of sound. Then within the cochlear duct there are hair cells with small protrusions that are connected. And as a certain area of the cochlear duct resonates, the fluid causes the hair cells to move and causes them to open. So as they are moved around by the fluid, cause openings that ions can travel through. So even though the endolymph already matches the intracellular fluid of the hair cells, there's still a driving force because the potassium ions are able to escape through leak channels at the back of the cell that lead to the scala tympani, which does not have as much potassium in its makeup. It is perilymph as opposed to endolymph, which resembles extracellular fluid. So as potassium moves into the hair cell, it is depolarized, which opens voltage-gated calcium channels that will release vesicles containing the neurotransmitter glutamate to a nearby neuron. So, calcium enters driving force from the scala tympani. It's what causes the driving force. Potassium, being a positive ion, depolarizes the cell, which allows calcium in, which releases neurotransmitters for a nearby neuron. So question 4, explain how the eye focuses light. Be sure to mention the role of the ciliary body and suspensory ligaments surrounding the lens. Describe the two types of photoreceptors and explain how they convert light into electrical energy. Explain how the center and surrounds cooperate in a visual field. So at a distance of 20 feet or more, the lens would be at its thinnest and the ciliary muscles relaxed. So the ciliary muscle is a sphincter, so its diameter is smaller when contracted and larger when relaxed. So at 20 feet or more, the ciliary is relaxed, suspensory ligaments tense, which pull the lens to be thinner. And then objects closer than 20 feet, the ciliary muscle must contract, lessens the tension on the suspensory ligaments, and allows the lens to thicken. And as the lens thickens, it bends light more, allowing objects that are closer to be focused more precisely on the retina. So, as an object is much closer, light needs to be bent more to be focused, as opposed to an object that's further away will appear smaller. So light from its ends do not need to be bent as much in order to converge on the retina. And the two types of photoreceptors are the rods and cones. Rods can only see in black and white, but are very sensitive to light and are responsible for our night vision. Many rod cells will converge onto a single bipolar cell. And many of these bipolar cells can converge. Let's see. And the bipolar cells can converge onto one ganglia cell. This allows many graded potentials from many rods to work together to build up and create an action potential. This allows our eyes to be very sensitive in low light situations, but because the visual fields are large, the image we put together in our brain is not a very sharp image. Cones, on the other hand, allow us to see color and are designed to deliver very clear images. So in the case of cones, not as many converge onto a bipolar, and not as many bipolar cells will converge onto a ganglia cell. This effectively creates more pixels for our brains to interpret. So sharper images are produced with cone cells. And cones detect light wavelength in the colors of red, blue, and green. And mixing these three inputs allows us to perceive about 1,000,000 different hues. Their visual fields are much smaller, so they give the brain more information to produce a sharper image. So for converting light into action potentials, it starts with a protein called opsin. See if this all fits. And opsin here contains a derivative of vitamin A called retinol. And in its unexcited state, retinol fits within a binding site in opsin. And when light of the right wavelength is absorbed by opsin, retinol goes through a conformational change in its hydrocarbon chain here and removes it from its binding site in opsin. That chink in the chain is removed, so that retinol no longer fits within opsin. So once retinol is removed, opsin is activated and its associated G protein, which removes the alpha subunit here. So before we continue, photoreceptors are unique in that they release neurotransmitters when they are not being stimulated. So when the cell is in the dark, cGMP here is bound to a cation channel that maintains the cell in a depolarized state. That causes the voltage-gated calcium channels to open and allow exocytosis of neurotransmitters. And then on the neuron of the bipolar cell, these neurotransmitters can be excitatory or inhibitory depending on the receptors on the bipolar cell that receive them. So the released alpha subunit from opsin goes on to activate the enzyme phosphodiesterase that will break down CAMP, or cGMP, into GMP, which removes it from its cation channel, closing it. Preventing these ions from entering will hyperpolarize the cell and cease its release of neurotransmitters. And so, onto the visual fields with surrounds and centers. So this is a visual field made up of many rods and cones and photoreceptors. So centers and surrounds work together to produce action potentials within a visual field. The center of a visual field can be activated or deactivated by the presence of light, just depends on the visual field. And the same goes for the area surrounding it, but they are always opposite. If this center is activated by light, the surround will be deactivated by light. And the activation of the center, whether or not it's activated by light or dark, doesn't matter. If it is activated, the center yields more action potentials. So these neurons from the center, from the surround, all converge on a bipolar cell. So they work together to feed a neuron. So center yields more action potentials, and this indifference of action potentials allows a sharp increase or decrease of action potentials if only a portion of the visual field is activated. So let's say we have a on center and off surround. So we'll split this up into 2, 6, 2. If the center is struck by light, it produces 6 action potentials, and if the surround is activated, in the absence of light, it produces 2 action potentials on each side. And so, if in complete darkness, the surround photoreceptors are on, producing a total of 4 action potentials, and the center is off or not activated, so it does not produce any. So in total darkness, we have 4. In partial darkness, if light only hits one part of this visual field, this one is activated in darkness, but is under light now, so this is not producing anything. The center is not producing anything because it is not in the light, and this produces 2. And then if we expand the light to include the center is now producing its 6 action potentials. This section of the surround is in light, so it is not producing, and this section is still in the dark, so it is producing. So we have a total of 8 action potentials in that case. And if it is in complete light, neither of sides of the surround are producing, and the center is. And so we have unique situations where different action potentials are produced depending on how much light and where that light is contacting this visual field. And so the extremes, the highs and the lows, are when light is partially hitting one of these visual fields. So this means that the edges of objects create unique signals on our visual fields, and so we create very sharp images due to the surrounds and centers of our visual fields activating and deactivating.